wine spoilage, and the emphasis is on using chemical and microbiological analysis to assess, or I suppose recognize, risky conditions in your process that can lead to spoilage. I think the slides are probably more geared towards red wine production than white, but there's a fair amount of overlap. This slide's pretty straightforward. Winemaking is just managing a biological process. Converting grape juice to wine involves, obviously, yeast and bacteria. And so your job as a winemaker, for example, is to shepherd the microbes appropriately, allowing the good microbes to do what you want them to do, and suppressing the bad ones and keeping them from doing what they want to do. So the concept of spoilage is typically broken into two versions, covert and overt. Another important concept on this slide is that spoilage can be caused by more than one microbe. And so we see this a lot, for example, in public health micro, where we like to assign a single disease to a single microbe. But in reality, when you dig into a wound or whatever, it's often many different kinds of microbes causing the damage. And the same thing happens in wine micro as well. Our sophisticated tools now are letting us see that there's a synergy by multiple kinds of microbes typically. And so in this case, this Venn diagram illustrates covert spoilage on the left here. And a lot of this covert spoilage is, as the name suggests, it's not detectable, but it's building up and things that are not easily detected with a instrument of some kind, or a high-level sensory analyst. But then as you reach this crossover point, you can start detecting some of this stuff using sophisticated machinery and potentially high-end, people who are sensory analysts. And then obviously we start getting into covert spoilage, where things become really obvious, things that you can see, smell, and taste, or the typical person could see, smell, and taste. This slide is an important one in the sense that it divides the process decisions into forensic versus preemptive. Now, obviously, hopefully, you want to be on the right side, the preemptive side, the side where individuals do monitoring and chemical and microbiological analysis. The idea here is obviously to catch problems before they happen, or before they get worse. And obviously on the left is the forensic side, where you have a problem, you lose a bunch of product, for example, and then you look backwards and try to figure out what went wrong. So from a winemaker's perspective, obviously preemptive screening is ideal. And even from a industry standard, for example, ETS Labs wants people to be more preemptive, because that means that they're paying more money for their screening assays and so forth. It's a win-win. Obviously, forensic is not a win-win. Somebody will lose in that when you have to look back on why things went wrong and you've lost a bunch of money. So that's the purpose of this slide, what the individuals at ETS have talked to me about. The majority of clients who are advocates of preemptive screening do so because of a major spoilage event. It's probably no different in anything. For example, if I've never had earthquake damage before, I'm less likely to buy earthquake insurance. So it's just a human condition that we all deal with. This slide is just sort of an overview of some of the specific defects broken into yeast and bacterial defects. And so things like ethyl acetate, for example, which is more of like a nail polish remover kind of nastiness that yeasts can produce through the metabolism of acetic acid and ethanol. Sulfides are these rotten egg smelling things, and that usually comes from amino acid catabolism. And I'll talk more about that, or at least I've talked more about that in the metabolism slides. Aldehydes, I've talked a bit about aldehydes, specifically acetaldehyde, which kind of has a tart green apple flavor, and you can imagine that that's kind of desirable at a certain level, but obviously you can get too much of it and it can be pretty gross. 4EP and 4EG are really important ones as well. 4EP has this guy here has sort of a medicinal wet Band-Aid flavor and aroma, and 4EG is kind of not so bad. It's kind of smoky, clovey, and so, but in the context of a wine, they're both disgusting. And I work with Brett, and Brett can make both of those compounds pretty efficiently. And then, of course, isovaleric. I've never smelled isovaleric, but I've heard it described as sweaty cheese, which is, again, pretty dang gross. But Brett also makes isovaleric acid. And bacteria do a lot of the same things. As you can see, there's a lot of overlap on the list, but you start seeing bacteria doing additional things like producing biogenic amines, which I'll talk about, and mousy off flavors. Again, you just go to PetSmart or whatever and wander around the where the mice and stuff are, and that smell that you get, that's that mousy kind of aroma. And obviously that's not ideal in a wine. And then certain, obviously VA, acidity, volatile acidity, and ropiness is an interesting one too, because it's more of a textural thing, where microbes can polymerize sugars like glucose and turn it and create a little bit of a slime, and so that creates a weird mouthfeel and almost grosses people out. But some of the groups of microbes, I've got it laid out here in a couple different groups, and so you have the heterofermentative lactic acid bacteria. So again, the lactobacilli, and then our malolactic organism, Coccus, falls into that. So again, heterofermentative just means that they make lactic acid, they'll make gas, and other end products from their fermentation. Whereas homofermentative means these are lactic acid bacteria that primarily just make lactic acid from fermentation. And then there's the non-lactics, like the acetic acid bacteria, for example, that obviously make acetic acid, and Brux will make the 4EP and 4EG that I talked about, and isovaleric, and Zygosaccharomyces as well, which I'll talk a little bit more about. These guys at the bottom producing potential extra fermentation products, Hansennia spora and Pichia. Pichia actually can make some 4EP and 4EG as well, and Hansennia spora can make ethyl acetate, which I've already talked about. Saccharomyces, I think one of its big issues is if it kind of hangs around in the bottle later around, you can get kind of fizz and zing in a bottle that where you don't want that. So that's the extra fermentation product typically that we worry about from residual Saccharomyces. So this slide's just looking at lactobacillus, and again produces large amounts of VA, particularly lactic acid, and also a lot of mousy type end products, these heterocyclic bases that I'll talk about. So again, they can be heterofermenters or homofermenters, and looking at them under the microscope is pretty diagnostic. They're gram-positive rods that form long chains, and they're big rods too. So these are organisms that are, if you start detecting high levels of VA, for example, you dip into your product and do a quick gram stain, and you see a lot of this, you can start a diagnosis pretty quickly. So this slide's just introducing you to the concept of biological amines and where these things come from. So typically they're produced from amino acid breakdown. And so in this example, this is ornithine, which is found in grapes. So it's present, and microbes are capable of producing foul products from it. So here's the traditional diagram of an amino acid. And so you have the amino group, the central alpha carbon, and then the carboxyl group. And microbes will produce an enzyme called decarboxylase, which can cut this carboxyl group off. And we end up with a biogenic amine. Here's the CO2 that gets emitted, and we end up with this biogenic amine. So in this particular case, if a microbe decarboxylates ornithine, you make this foul material called putrescine, which, as the name suggests, gives the aroma of putrid, rotting meat kind of smell, which is pretty disgusting. These are just some structures to show you. Here's the structure of putrescine, for example, this linear molecule. And here's another fun one called cadaverine, which when a microbe decarboxylates lysine, it can convert lysine to cadaverine, which cadaverine is one of the reasons why dead bodies smell the way they do. But something that's more likely to happen, or maybe as likely, if not more likely, to happen in wine are the formation of histamines and tyramines. So tyramine is the decarboxylation of tyrosine, the amino acid tyrosine. And tyramine can actually influence and cause a weird irregulation of your blood pressure. And so that could be a problem, obviously, if you have an elderly, for example, wine drinker partaking of your wine and there's elevated levels of tyramine, you could potentially kill them. And similarly, histamine, that's the decarboxylation of histidine, the amino acid histidine, by a microbe. And so the microbes producing histamine can cause allergic reactions in the people that drink your wine. Obviously, histamine, hopefully that jogs something in your memory. Antihistamines are things that we take during allergy season. And so obviously, if your wine contains elevated levels of histamines, that might be a problem. So some of these, some of the bullet points here, I just wanted to go through obviously biogenic amines can occur naturally in grapes, and certainly if the biogenic amine itself doesn't occur naturally in grapes, the starting material, the amino acids, definitely can. And so again, they result from yeast breakdown of proteins in wine. So a yeast can break down proteins, releasing free amino acids. Then the microbes, the bacteria particularly, will grab those free amino acids and start decarboxylating them to produce these various kinds of biogenic amines. It can also result from breakdown of proteins in wine during malolactic conversion. And so bacteria can actually do this too. They can actually break down the proteins and in turn decarboxylate them. And so Enococcus, our malolactic organism, can actually do that. In some cases. The result is typically an overt moldy tuna or rotting meat kind of smell. There's also the potential for a covert if it's not so intense. If you don't get up to that sensory threshold, you can often get a weird loss of aroma type of spoilage if certain kinds of biogenic amines are formed. Usually they're used as an indicator for poor sanitation, and I put that in here as a European Union limit of roughly 10 parts per million or 10 milligrams per liter. An ETS survey, I thought this was interesting data that they conducted on 284 samples of different kinds of wines in California, Oregon, and Washington, showed that a pretty significant number of them, at least nearly half of them, exceeded the higher end limit of 10 parts per million or 10 mgs per liter. Same thing. That's proposed by the EU. But I should also say that we know that white wines have lower levels generally than of biogenic amines than reds, but we also know that not all biogenic amines should be treated equally. So, for example, histamine, I think they say maybe 2 to 10 milligrams per liter, or again parts per million, is acceptable for histamine, but for tyramine, I don't know. I've read a lot of literature that suggests that 10 might not even be safe. Again, when we're talking about fluctuations in blood pressure, that's dangerous. So as far as prevention goes, certainly using a commercial malolactic strain such as Enococcus eni, that is a commercial strain that's been vetted and is known not to create biogenic amines, because obviously that's ideal. Some people like to purify and isolate their own microbes, and they don't vet them for certain things such as biogenic amine production. One thing is using yeast strains that leave lower levels of amino acids. So you want your primary yeast, your Saccharomyces, to be really good at soaking up amino acids and catabolizing them, and basically just taking them away from the bacteria. And obviously DAP, if you're using diammonium phosphate, usage can increase biogenic amine formation. It shouldn't, obviously if you're overusing it, I should say. It could cause problems, because DAP is a nitrogen source, and the microbes can take that nitrogen and use it to make amino acids. And obviously, more amino acids equals more biogenic amine production. Certainly, limiting contact time with skins is a big one as well. A lot of people like to use an extended maceration. One of the things that it does is polymerize tannins and sort of make these larger tannin type molecules, because the larger tannins tend to be less bitter than smaller individual tannins. And so longer maceration, makes a lot of sense. But you could also increase the output of arginine, right? And so, as I've already shown you, that arginine can be converted to ornithine, and in turn, ornithine can be converted to putrescine, which is the biogenic amine. One of the other things that happens in this type of metabolism, though, is arginine conversion to ornithine can also result in the formation of urea production. So the microbes will make a little side urea, and urea can actually combine with ethanol to make ethyl carbamate, which is, I don't know if there's any sensory to that, but I definitely know that it's toxic. And I don't know what the threshold level is, but you certainly don't want any kind of known carcinogens floating around in your wine either. So ethyl carbamate production from arginine metabolism is a potentially bad outcome as well. The other issue is extended contact time with lees, and so that can certainly increase biogenic amine formation. One of the reasons for that is that we know that dead yeast, which basically is what lees is, or lees are, is basically just a bunch of dead yeast that are releasing sugars and amino acids and fatty acids. And obviously it's a good thing, right? Because it adds flavor complexity and aroma to a wine. But that release of amino acids that I just talked about can increase biogenic amine formation, especially in higher pH wines. We found that microbes like lactobacillus and pediococcus and even enococcus can grab a hold of those amino acids leached out of the lees to increase biogenic amine formation.